Hey everyone, this is going to be a quick walkthrough of the sample assessment for the new ChemBio genetic variation standard. Annoyingly, it's only the one question with part A and B in. That's all we've got to go off of with regards to our prep for the real exam on Tuesday. Saying that, the old standard that this one is more or less based on is very, very similar. A lot of the content's the same, so if you've been using the old standard study guides like we have in class, you'll have seen that a lot of the content is the same. Genetics is genetics, whichever way you examine it. The question styles, there are a few that I've seen that are slightly different focuses. So if you do come across those, just bear in mind that it may not be representative of what you see this year. It's a good place to practice the content and your understanding, but the question styles may be slightly different. Okay, we're going to jump straight in and do as much as we can with what we've got. So, we are told that we are going to be dealing with the allele for HIV resistance. The human immunodeficiency virus, HIV, is a virus that infects the white blood cells of the human immune system. Individuals who are normal CCR5 gene are susceptible to HIV infection. Mutations can occur in this gene, which results in a mutant CCR5 allele. Individuals who are homozygous for the mutant allele are resistant to HIV, while individuals who are heterozygous are still susceptible to HIV infection. What I've done down here is just using the H for you're susceptible to HIV and lowercase h as the mutant allele. I've just gone through and said non-resistance is dominant, therefore you have to be homozygous recessive in order to have the resistance, just because it sort of breaks this down into 3 quick and easy points rather than all these words. Before I jump into the actual questions, just very quickly, to make sure we're clear on what's going on, the human immunodeficiency virus, HIV, is a virus that causes AIDS. So this disease, this pathogen, this virus, if left untreated, leads to AIDS, which is the autoimmunodeficiency syndrome, where you essentially have a compromised immune system for life. So it's not that disease in itself is particularly damaging to your body, but it shreds your body's defenses, its immune system, so it can no longer defend itself from any other diseases. So if this mutant allele does provide resistance, this could be a very, very important mutation going forwards. And if we were able to see a long way into the future, it may be that the prevalence of this mutant allele becomes quite common throughout humanity, because AIDS is essentially untreatable. You have to manage it very carefully. So, the other thing, is genes. If you see genes given with these sorts of names, numbers and letters like this, because there's so many different types of genes across all organisms, they have to use codes like this. So you haven't got to memorize them. This doesn't mean anything, doesn't tell you anything. This is just the name of it, okay? Now, in general, there's a general rule with these sorts of assessments. It seems to be if you just give definitions or explain what the key terms mean in the question, you pick up achieve points, usually about 1 per key term, particularly the ones that it tells you to include or it suggests that you talk about. For merit, you're then starting to link that to the general concepts at play as to how the genetics cause phenotypes or mutations, that sort of stuff. The excellence comes from doing all that thoroughly and completely, and also linking it back to this context, okay? So in theory, you don't need to actually worry about this. If you're just gunning for achieved, you just essentially can give definitions and try to answer the actual question, and you'll probably pass. If you're aiming for merit and excellence, you should be trying to tackle the actual context and the questions themselves. So we'll jump straight in. Question 1. Explain how a mutation in the CCR5 gene is able to provide resistance to HIV. In your answer, include the terms gene, allele, phenotype, and DNA. So I'm just going to focus the camera. So these are our key terms that I'm going to go through and define to begin with. As I do so, I'm going to start crossing stuff off just so that I know what I've done. Particularly for the longer questions, when you've got a whole side of A4 to do it, you might lose track of what you're doing. So we'll talk about how you might plan your answer as well for part B, the longer sort of questions. So I'm going to start with DNA first, because that's the most basic component of this standard. So we're just going to define very briefly what DNA is. a chemical that stores genetic information in its order, because there's not a lot of space in this sample. Sorry, I am going to try and write on the edges, which obviously you shouldn't do. In its order of bases, or the sequence of bases. There's not one specific way to phrase it. The mark scheme, I think, just says defines it or describes it. So you've got a bit of leeway with how you describe it. Key point is it stores genetic information in an order of bases. That's important because we're discussing a mutation here. So if you're talking about mutations, you should be thinking about the order of bases that are going on. The next term we're going to do is a gene. It makes sense to do DNA first because we're going to reference DNA in our definition of gene. A short section of DNA that codes for a specific protein. Okay, and again, we're mentioning that it's a short section of that DNA code because we have a mutation coming up. Then we have an allele, which is nice and straightforward. An alternative form of a gene, usually with a different, and again, I'm going to squeeze it on the end, which you really shouldn't do, with a different phenotype or physical expression of the gene. Sorry, or the allele, my bad. Easy to mix them. So now that I've referenced phenotype, sorry, it's the end of the period. Phenotype, I don't think anyone's in this classroom if I suddenly stop mid-sentence because the class walks in. Phenotype. A phenotype is, quite simply, the pH tells you it's physical. This is the physical expression. I'm sorry for any noise in the corridor, and right next to the door as well. The physical expression of the genotype. If we have more space, we might try and describe what genotype is very quickly. Because the question doesn't really ask for it, I'm not going to, can't be bothered. So we've got our 4 definitions. That's always a good place to start. These are just from memory. You haven't got to think about HIV or the context at all. You just rattle them off straight away. And in theory, you should get a number of achieve points for doing this. Just quickly glancing ahead at the marks, I think you can get 3 points. I'll explain it later. 3 of these definitions get you an achieve mark. So A3 is achieved already here. Now, just quickly, the actual question was to explain how a mutation in the CCR5 gene is able to provide resistance to HIV. If the order of, first of all, sorry, I'm describing what a mutation is. It doesn't make sense to talk about mutations and their effects without saying what it is, just in the same way as we've defined all these key terms. If the order of bases in the CCR5 gene changes, we get, sorry, I missed that word. If the order of bases in the CCR5 gene changes permanently, we get the mutant allele. In order to have the resistant phenotype, I'm running out of space, an individual must be homozygous. Balls, I think there's a class about to come in. Homozygous recessive. Sorry, I'm just going to pause. Sorry, they were in the wrong classroom. So, first of all, in the real exam, you shouldn't have this little space. I think this is just very short because it's the sample. They haven't really formatted it appropriately. Now, as I said, with these definitions, you get your achieve points. You got the definition, the achieve points if you defined mutation, DNA or chromosomes. Sorry. Okay. So you get your definitions of. It's a bit annoying because the mark scheme is awarding credit for both parts A and B. So it's not just limited to part A. In this, you would have got an achieve point for DNA, describing what it is. You had to do mutation, changing the order, which is what we said. Genes, alleles, phenotype, and genotype. We will probably talk about genotype in the second part anyway, so that's fine. For merit, you had to talk about the fact the individual must have 2 copies of the resistant allele, the homozygous recessive one, in order to express it, as well as explaining what a mutation is. Okay. That was the one merit point you can get, so I don't think you can get any excellent points in this part. Now, for part B, again, we have not a lot of space. This is supposedly where you can earn all your excellence points, so I may have to start using this book in a sec. But we'll see how we get on. So we are discussing how the CCR5 gene mutation can be passed on to the next generation, leading to genetic variation in population. In your answer, you should consider how sexual reproduction, including meiosis, contributes to genetic variation, the advantages of genetic variation in a population. Okay, so what we're going to do, just on the side here, I'm just going to sort of jot down a few key checkpoints for things that I want to try and talk about based on the actual question and these suggestions as well. So, if we're talking about inheriting mutations or passing on mutations, we're going to talk about fertilization plus gametes, because the mutations have to occur in the gametes. I need to make sure I discuss that part as part of my fertilization. The sexual reproduction, also including meiosis. We could then talk about our sources of variation, such as assortment, independent assortment, plus crossing over. Leading to genetic variation. So the advantages of it, advantages of variation. And then because it's asking us in the actual question to discuss how this can lead to variation in the population, we're going to sort of discuss how that gene might propagate, or the mutation might propagate over many generations. We're going to see it's not a description of natural selection, but it's got a lot of similar sort of terminology and ideas to discussing natural selection. So I'm going to say future generations, in brackets, nat cell. I'll explain that bit in a sec. So, as I go through my answer, hopefully I can tick off these bits as I do them, just so I make sure I have some sort of structure to my answer, and I'm trying to cover all the bits across the question and the suggestions. So, first of all, we're going to say, with regards to the fertilization and gametes, for the mutation to be passed on, it must occur in the gametes, DNA. This is because during sexual reproduction, two gametes fuse together and combine genetic material or genetic information to fertilize one another. The offspring will contain the mutation in all of its cells due to mitosis. Sorry. So you need to have the mutation present in the DNA of the sperm or the ovum. If you don't, if the mutation occurs, let's say in the skin on the end of your fingertip, that bit of DNA does not get passed on to any offspring. So it has to occur in the sex cells. This little bit here discussing the mitosis is very quickly, because when the zygote starts developing and growing and doing cell division by mitosis, because it's mitosis, all of the DNA is identical in all daughter cells, and so everyone will gain that mutation, or will have that mutation. I have now discussed fertilization and gametes, so I can take it off. I can almost tick off this first bullet point, but we have discussed passing on. I'm not going to tick that because it's going to mess up the bit above it. So, the offspring will contain mutation laws in its cells and mitosis. Now we're going to talk about where the genetic variation comes from. So, meiosis. Sorry, some loud kids outside. One sec. Sorry about that. Meiosis contributes to genetic variation as every gamete is genetically unique. I'm just going to very briefly, because it's not really the main part of this, it's not asking what are the sources of variation. I'm just going to very quickly name the different sources of variation, and just as a justification for my previous point. Meiosis contributes to genetic variation as every gamete is genetically unique. This is due to them being haploid, exchanging material between homologous chromosomes, which is crossing over, and chromosomes being sorted into daughter cells at random. Random, and I am running out of space very quickly. Sorry, just to catch you on the end of the page. So, don't need a lot of detail here. I've just discussed the sources of variation in meiosis, and so I can take off that little section there. So now my next step that I told myself I was going to do is to discuss the advantages of variation. Because I have reached the end of the paper, I'm not going to start here. I'm going to use what I think is an abandoned book. I can look either way. I'll just pop this, hopefully, on the top there. How am I going to do this? No, I'm going to rewrite my remaining two checkpoints on the left. My add of variation, sorry about this. Again, I didn't make the sample assessment. Future generations is my other thing to talk about. Now we can make a start. So with the advantages of variation, this is the idea of how you can be resistant to diseases. And obviously in this case, we are talking about the resistance to HIV. So significant. The advantages of genetic variation are that a population, sorry, it does say population. I'm going to unfold the book actually, so it doesn't hide it in the corner so much. You see that? A bit glary. Population. The population has a range of traits. Okay. This means the population, not necessarily the individuals, is more likely. It's not a guarantee, as the dinosaurs found out, but it does mean that it is more likely that they can adapt to changing environments, such as an asteroid, or the one that's relevant to this context, or have resistance to diseases, such as HIV. Now, this is where it gets a bit weird because obviously humans, with our technology and medicine and our understanding of the natural world, we have ways that we can sort of beat this natural rule that occurs for most organisms, or will have occurred for humans for many, many years. So yeah, doesn't really make sense for us to talk about this now because obviously, like we know how things work, so we can make adjustments to our lives to help this not occur, this not spread, even if we're resistant or not. But yeah, so we talked about the advantages of variation. Then for future generations, this is where we're going to talk about the natural selection sort of stuff, because the question was asking, how does the gene get passed on to the next generation, leading to genetic variation in the population? So over many generations, individuals with the resistant phenotype are more. This is why you do this is why I had the natural selection bit, sorry, before, because if they have the resistant phenotype, are more likely to survive, and this is where I'm going to reference my HIV context to try and get those excellence points, are more likely to survive HIV infection. And the more they survive, if they are resistant to the virus, I'm now going to expand on that. If they're resistant to the virus, people are more likely to survive until adulthood. Although technically speaking, this in the wider animal kingdom, plant kingdom would be sexual maturity, but the same idea basically. Are more likely to survive until adulthood, and therefore reproduce. Therefore, the mutant allele is more likely to be inherited by offspring. Meiosis is more likely to be inherited by offspring. So over time, or many generations, being a bit more specific, over many generations, resistance to HIV should become more prevalent. Does that say prevalent? Not very well. Prevalent, or it's more common, it's more frequent, or more individuals have this trait. Resistance to HIV should become more prevalent in the population. So, I've discussed my advantages of variation, I've discussed my future generations for my natural selection sort of stuff, talking about surviving till adulthood, therefore reproductive age, therefore passing on traits, therefore the population should increase the number of individuals with the resistant mutation. Now I'm just going to quickly go through it and check the question. I haven't missed anything obvious, and we'll look at the mark scheme together. Have we discussed how the CCR5 gene mutation can be passed on? Yes. Have we discussed how it can lead to genetic variation? Yes. Have we considered sexual reproduction, including meiosis, contributing to variation? Yes. Have we discussed the advantages of genetic variation in the population? Yes, we have. So in theory, we've got all the marking points, but I seem to recall when I looked at this the other day, there was something a bit weird I didn't think made sense to put in here, but we'll go through and look at it. So I'm looking at both parts together now. For the achieve, this is where we were discussing, if you describe what a gamete is, so gamete having, did I say they were haploid? Yes, I did. Thank God. So we've discussed gametes, we discussed fertilization. We haven't described meiosis. This is what it was, sorry. I don't think you need to, it doesn't mean describe the actual process. I think it means describe the sort of outcome. So this idea of you having genetically unique gametes that are haploid that fuse together to fertilize together and can experience crossing over, independent assortment, that sort of stuff. I may be wrong, but the question, when it gives an example, doesn't describe what meiosis is. That bit on the left. So yeah. For merit, we had to explain how the DNA can code for resistance, and explain how all these various bits and bobs relate to each other. So DNA relating to a gene, relating to an allele, relating to a phenotype, which is what we discussed sort of in part A and B. This is why the mark scheme allows you to choose from both questions. We had to discuss the effect of the mutation, the fact that you would be, in some vague terms, more resistant to the virus in a population and more likely to survive. For merit, you haven't got to go into much detail, just give the overall point that the mutation should increase your survivability because you're less likely to catch HIV. The last merit point, is you have to explain the inheritance of an allele in a parent's gamete passed on following fertilization, which is what we talked about down here. So I think I've got all three merit points. Sorry, it's a lot to read very quickly. I think I have, yes. Then for excellence, we are talking about the phenotypical advantage to inheriting or developing the mutation with a higher chance of surviving to reproductive age and therefore passing the mutation on. So that last excellence point is essentially just this discussion, this natural selection style discussion or description here. Now, if you have worded stuff slightly differently, you're not sure if your bullet points are meeting their bullet point to match your answer, the big paragraphs on the left give you an example of how they might expect you to describe it or explain it. The wording will be different, the order will be different. But yeah, as long as the general focuses and the general descriptions are there, I think you should be quite okay. I actually can't see any mention of crossing over assortment, so I don't know why it specifies meiosis. I think that specification of talking about meiosis was just the haploid gamete part during fertilization. I hope this is helpful. It's not very clear just because we have so little to go off of, and we've had, as teachers, quite little, guidance on what sort of things will be examined content-wise and how they will be examined. As you can see, we've only got 2 questions. We haven't been given anything else. It looks very similar to the old standards. That would be a good place to start if you want to do some more practice. I'll put the link in the description. Give it a shout in classroom or email if there's anything else you need or any other particular things you want me to go through. Or any other questions from any other sources like the SciPads that you want gone through. I won't be able to put them on YouTube publicly, but I can put them on our Google Classroom at least. So give us a shout and check out the link in the description if you wanted to see the old standard resources from No Brain Tutorials. There are loads on there and they are good. Oh, and I'll do some flashcard links as well.